The impossible has happened. A creature long gone, vanished from the earth for millennia, now walks among us once more. The dire wolf, the legendary predator of the Ice Age, has been brought back from extinction. This astonishing feat, once confined to the realms of science fiction, is now a reality, raising profound questions about our relationship with the natural world and the very essence of life itself. For centuries, the dire wolf was known only through fossilized bones, silent remnants of a bygone era. They whispered tales of a time when giants roamed the earth, painting a picture of an apex predator that ruled the Pleistocene landscapes. These were not simply larger versions of the wolves we know today, but a distinct species, Canis dirus, more powerfully built and adapted for hunting the megafauna that shared their world. The very idea of bringing back an animal from extinction seems like something out of a fantastical novel. Yet, here we are on the cusp of a new era, where the line between science fiction and reality is becoming increasingly blurred. The implications of this breakthrough are far-reaching, touching upon ethics, ecology, and our very understanding of life and death. The return of the dire wolf is not just a scientific marvel, it is a powerful symbol of both our potential and our responsibility. As we stand at this crossroads, we are compelled to ask ourselves, what does it mean to bring back the dead, and what will be the consequences of our actions? The resurrection of the dire wolf was made possible by groundbreaking advances in the field of genetics, particularly in our ability to extract, sequence, and manipulate DNA. DNA, the blueprint of life, holds the instructions for building and maintaining an organism. Even though an organism dies, its DNA can persist for thousands, even millions of years, under the right conditions. The first hurdle in resurrecting the dire wolf was obtaining viable DNA samples. Fortunately, numerous well-preserved dire wolf fossils have been unearthed from places like the La Brea tar pits in Los Angeles, California. These tar pits acted as natural traps, preserving the remains of countless animals, including dire wolves. Extracting DNA from ancient specimens is a delicate and painstaking process. Over time, DNA degrades and becomes fragmented. Scientists use sophisticated techniques to extract these fragments and piece them together like a jigsaw puzzle, reconstructing the dire wolf's genome. This process, while remarkable, is not without its challenges. It requires meticulous care to avoid contamination and to ensure the accuracy of the reconstructed genome. The successful extraction and sequencing of the dire wolf's genome was a momentous achievement. It provided a glimpse into the genetic makeup of this extinct predator, revealing fascinating insights into its evolutionary history, physiology, and relationship to modern-day canids. It also provided the crucial genetic blueprint needed for the next stage of the resurrection process, cloning. With fragments of DNA extracted from well-preserved fossils, scientists faced the daunting task of piecing together the dire wolf's genetic code. Imagine a jigsaw puzzle, not with 1,000 pieces, but with millions, each one representing a tiny fragment of DNA. Sophisticated computer algorithms, designed specifically for this purpose, meticulously analyze the overlapping fragments, gradually assembling them in the correct order based on their sequences. This was not a simple linear process, but rather a multi-dimensional puzzle, requiring the identification and placement of genes, regulatory elements, and other crucial components of the dire wolf's genome. The process was further complicated by the fact that DNA degrades over time, leaving gaps and errors in the genetic code. To fill these gaps, scientists turned to the genomes of closely related species, such as the grey wolf and the domestic dog. By comparing the genetic sequences, they could infer the missing pieces of the dire wolf's puzzle, effectively filling in the blanks of its genetic blueprint. The complete genome of the dire wolf is more than just a scientific curiosity, it is a treasure trove of information. It allows us to delve into the evolutionary history of this fascinating creature, tracing its lineage back through time and understanding its relationship to other canids. We can study its genes to learn about its physical characteristics, its adaptations for hunting, and even its behavior. Cloning, once a concept confined to science fiction, has become a reality in recent decades. However, cloning an extinct animal like the dire wolf presented unprecedented challenges. Unlike cloning living animals where fresh DNA is readily available, Resurrecting the dire wolf required overcoming the hurdles of degraded DNA, finding a suitable surrogate, and navigating the complexities of embryonic development in an extinct species. The fragmented nature of ancient DNA posed a significant obstacle. Scientists had to develop innovative techniques to repair and amplify the degraded DNA fragments, ensuring the integrity of the genetic code before attempting to create a viable embryo. Finding a suitable surrogate mother was another hurdle. While the grey wolf might seem like the obvious choice, the evolutionary distance between the two species meant that a grey wolf's womb could not support the development of a dire wolf embryo. After much research, scientists identified a promising candidate, the African wild dog. Although not as closely related as the grey wolf, the African wild dog possesses a similar reproductive physiology and gestational period, making it a more compatible surrogate for the dire wolf. Through a combination of cutting-edge gene editing and assisted reproductive technologies, scientists successfully created a viable dire wolf embryo and implanted it into a carefully selected African wild dog surrogate. The world watched with bated breath as the surrogate mother carried the dire wolf pup to term, marking a pivotal moment in the history of science and conservation.